In March of 2026, scientists sequenced the DNA found on Christianity's most famous relic, the Shroud of Turin. And, well, they uncovered a data point that completely broke the internet. I mean, almost 40% of the identifiable human genetic material on that cloth was from the Indian subcontinent.
Which, yeah, as you might expect, caused an absolute media meltdown. People saw the words Indian DNA and immediately jumped to, the most sensational conclusions possible about the historical Jesus.
Right. Welcome to the deep dive. Today, we are looking at the actual science behind those massive headlines. We're pulling from the 2026 genomic research papers, archaeological data, and the cultural history surrounding this artifact. And the mission for us today is to untangle the biological history of the physical linen for the genetic identity of the historical figure associated with it.
Okay.
Let's unpack this. Think of the shroud not as a photograph of a single person, but as a heavily stamped biological passport.
Oh, I like that.
Yeah, it is a fabric document that has quietly recorded a microscopic visa from every single stop on its ancient, chaotic journey.
That is a phenomenal way to frame it. I mean, to understand what this artifact actually is, we really have to look past the religious icon and analyze the cloth as an archaeological survivor. We are talking about the true physical interconnectedness of the ancient world.
Absolutely. So let's jump straight into that explosive 2026 data. Because to figure out how the media arrived at the Indian DNA bombshell, we have to look closely at the methodology. This was a seminal Bioarc-6 study led by Professor Gianni Barcacci at the University of Padova, and they weren't, chopping up the shroud with scissors to get these samples.
Oh, no, far from it. They actually utilized dust and organic residue that had been vacuumed off the shroud decades ago.
Wait, vacuumed off?
Yeah, during the 1978 Shroud of Turin research project, which is usually called STERP, back then researchers ran specialized vacuums and sticky tape over the linen to collect loose microscopic debris.
Wow, okay.
And that dust basically sat in storage until the Barcacci team applied something called next-generation sequencing, or NGS, to it.
And just so we're all picturing this correctly, how is next-generation sequencing different from, say, the DNA tests we might have taken a decade ago?
So older DNA sequencing was highly targeted. It was like going into a library to look for one specific sentence in one specific book. You had to know exactly what you were looking for.
Well, right, very narrow.
Exactly. Next generation sequencing is like scanning every single book in the entire library simultaneously. It reads millions of DNA fragments in massive parallel batches. And for this study, the Padova team specifically targeted mitochondrial DNA, or mtDNA.
Which is incredibly smart for ancient forensic work, just for anyone listening who hasn't taken biology in a minute. Mitochondrial DNA is basically the genetic material found in the energy centers of our cells. And it's passed down directly from your mother.
Right.
But the reason it's so crucial for an ancient artifact is its sheer resilience. I mean, a human cell only has one nucleus, but it can have hundreds or even thousands of mitochondria. So when biological material degrades over centuries, your odds of finding intact mtDNA are exponentially higher.
Yeah, and that structural resilience allowed the Padova team to open up a literal biological encyclopedia trapped within the weave of the linen. They mapped the DNA into haplogroups, which act like ancient genetic barcodes.
Yeah, barcode.
Yeah, a haplogroup is essentially a specific mutation that tells us what part of the world a particular maternal lineage originated from. And when they tallied up the identifiable human DNA, the breakdown was stunning.
Let's hear it.
So 55.6% of the DNA was Near East or Levantine, featuring markers like the H33 and ROA haplogroups, and less than 5.6% was Western European.
But then comes the data point that hijacked the news cycle. There was a massive chunk, between 38.7% and 40%, that traced straight back to South Asia, specifically the Indian subcontinent, featuring the M&R haplogroups. Yeah. Almost half the identifiable human DNA on this Mediterranean relic has a South Asian origin.
It is a staggering proportion. It really is.
I have to stop and push back here, though. It's just following standard logic. Right. If you find a burial cloth draped over a specific historical figure, and 40% of the DNA on it is South Asian, why shouldn't you naturally assume the person wrapped inside it was South Asian? I mean, why wouldn't that be your first scientific conclusion?
It is a totally fair question, and honestly, it's the exact trap the initial news reports fell into. What's fascinating here is what geneticists call the cornucopia effect.
The cornucopia effect.
Yeah. When the Barcacci team analyzed that NGS data, they didn't find one cohesive Indian genome. They found a shattered mosaic. This DNA belongs to a multitude of different individuals. It's an aggregate.
Oh, I see. So it's not a single biological smear left by one body. It's the microscopic fingerprints of a crowd.
Exactly that. Just imagine the sheer volume of hands that have touched this cloth over the centuries. Weavers, merchants, priests, pilgrims. Every time someone speaks over it, touches it, or kisses it, they shed skin cells and saliva. The researchers were very explicit that it is scientifically impossible to isolate one original genome among the thousands of biological contributors on that fabric.
Okay, so if that massive 40% South Asian genetic signal doesn't belong to the man in the shroud, we have to figure out how it physically got onto a Mediterranean relic in the first place, which leads us into a completely different kind of history of ancient global supply chains.
Yeah, the researchers put forward what is known as the manufacture hypothesis. They propose that this enormous Indian genetic signal is simply the result of how and where the fabric was made.
Makes sense.
Right, because in the first century, the Indus Valley, specifically the region of Sind, was an absolute global powerhouse for high-quality linen and yarn production.
And the linguistic clues here are staring us right in the face. The word shroud itself comes from the ancient Greek word sindon. Scholars have long theorized that sindon isn't just a generic term for cloth. It literally refers to fabric imported from the Sind region. It's named after its origin.
There is also robust textual evidence for this. Ancient rabbinic sources routinely mention Hindoyim.
Hindoim.
Yeah. These were highly prized, luxury imported linens traveling from the Indus Valley straight into the Roman-controlled world.
But linguistics can only take you so far. The 2026 study backed this up with hard botanical evidence, which I find incredible. They didn't just find human DNA from South Asia. They found plant DNA woven into the same fibers.
Yes, they did.
Specifically, Vigna unguiculata, which is the common cowpea, along with other plants from the Fabaceae family that are native to South Asia.
And we really have to think about the mechanics of ancient textile production to understand why that matters. Turning flax into linen is a brutal, messy agricultural process.
Oh, yeah.
You have laborers retting the stalks, scutching the fibers, beating them, and spinning the yarn. These workers are eating local food, sweating, and working in open fields where local crops are flowering. Microfragments of local plant matter get physically beaten into the cellulose structure of the linen.
So the co-occurrence of Indian human DNA and Indian plant DNA strongly implies that Indian laborers grew the flax, processed it, and wove it. Think of it like finding a modern Made in Taiwan tag on an antique jacket. That tag tells you about the complex global supply chain that manufactured the object. It tells you absolutely nothing about the ethnic heritage of the person who bought the jacket and wore it.
That reframing changes everything. The Indian DNA is the signature of the artifact's creation, and it paints a vivid picture of a first-century Judea that was thoroughly plugged into a massive, sophisticated global economy via the Silk Road and Roman maritime routes.
Right.
If a high-status individual was buried in Jerusalem, it makes total economic sense that their followers would wrap them in the absolute finest luxury goods available, which at the time were imported from India.
But the shroud didn't just capture DNA at its point of manufacture. As it moved across continents over 2 millennia, it acted like the ultimate biological sponge. And this concept of biological accretion heavily complicates how we date the artifact.
Oh, absolutely. Environmental DNA, or eDNA, is relentless. The cloth is a magnet for its surroundings. When the Padova team read the data, they found halophilic archaea. These are highly specialized microorganisms that only thrive in extreme salt environments.
How does something like that embed itself in linen?
Through prolonged environmental exposure. If the cloth was stored near the Dead Sea, for instance, the highly saline moisture in the air permeates the fabric. The salt crystallizes, trapping those local microbes deep within the weave. But it doesn't stop there. The researchers found marine life DNA like Atlantic cod and gray mullet. They found genetic markers from domestic animals, cats, dogs, horses, pigs.
And the ultimate proof of this continued biological absorption is the presence of New World plant DNA. They found traces of potato, tomato, and maize.
Which is wild.
It is. Potatoes and tomatoes were completely unknown in Europe or the Middle East until after the late 15th century. If their DNA is on this cloth, it proves the linen was actively pulling in environmental markers well into the modern era. I mean, even the 1978 Stirpe team inadvertently left their own modern human DNA on it.
Which brings us to the most contentious part of shroud research, the dating.
Here's where it gets really interesting. because all of this biological accumulation creates massive friction with the famous 1988 carbon-14 radiocarbon dating test. That 1988 test concluded the cloth was manufactured between 1260 and 1390 CE. It basically declared the shroud a medieval European forgery.
This raises an important question about how we interpret single-point testing on a highly contaminated object.
I try to picture it like attempting to date a 2,000-year-old oil painting. But imagine someone has been spraying a fresh layer of clear coat on that painting every decade for 1,000 years.
It's a great analogy.
Right. If you take a tiny snippet of the canvas and run a carbon test, you aren't just testing the original oil paint. You are testing the aggregate of all those clear coats. You're burning 14th century bacteria, 16th century tomato pollen, and 20th century skin cells right along with the original fiber.
And that biological sludge heavily skews the radioactive decay reading forward in time. The 2026 genomic data fundamentally challenges that medieval European timeline. Let's look at the H33 haplogroup they found.
Yeah.
That specific genetic marker is prevalent in the Druze population of the Levant. The Druze are famous for practicing strict endogamy, meaning they have married exclusively within their own closed community for centuries.
And by keeping their community closed, they preserved a very pure, ancient Near Eastern genetic signature.
Precisely. Finding a robust H33 signal embedded in the shroud points to a genuine ancient Middle Eastern history. The vast genomic diversity on this cloth spans India, the Middle East, and North Africa.
Which takes time to build up.
Right. An object requires a much longer exposure period than a few centuries sitting in cathedrals in France and Italy to accumulate that specific global footprint. It hints heavily that the carbon-14 sample taken in 1988 was either incredibly contaminated by biological material or came from a heavily handled, repaired section of the cloth.
So the metagenomic camp is essentially telling the radiocarbon camp that their timeline doesn't match the biological reality of the fabric. But this 40% Indian DNA figure leads us into a highly sensitive area.
It does.
Because despite the deeply logical ancient trait explanation we just discussed, those headlines immediately fueled a revival of an alternative historical theory about Jesus' ultimate fate. And we need to look at this objectively against the actual genetic landscape of the first century.
You're referring to the Rosa Ball tradition.
Yes. The 2026 data breathed new life into a theory initially popularized in 1899 by Mirza Ghulam Ahmad. He claimed that Jesus actually survived the crucifixion, traveled east along the Silk Road, and lived out his final years in Kashmir, India.
It is a deeply complex cultural tradition centered around the Rosa Ball Shrine in Srinagar, which houses the tomb of a figure known as Uz Asaf.
And the circumstantial elements people point to are fascinating. Proponents argue that Uz Asaf translates to leader of the healed, or acts as a linguistic corruption of Isa, the Islamic name for Jesus.
There are physical anomalies at the shrine as well. Islamic burials dictate specific directional alignments toward Mecca, but this particular tomb is aligned on an east-west axis, which proponents argue matches ancient Jewish burial customs.
Visitors also point out carvings of footprints at the shrine that appear to depict crucifixion wounds. Additionally, local Pashtun and Kashmiri traditions feature communities referring to themselves as the Bani Israel or the Children of Israel. There's a rich, undeniable cultural tapestry there.
And that tapestry led researchers, most notably an American author named Suzanne Olson, on a decades-long quest to find what she called Jesus DNA in India. She wanted to establish a genetic link to the Desposini.
And just to clarify for everyone, the Desposini refers to the purported blood relatives and descendants of Jesus' family.
Right, exactly. She petitioned to extract DNA from the Rosa Ball tomb, but she ran into massive, understandable roadblocks. Local caretakers fiercely blocked exhumation attempts, citing profound religious objections and the risk of desecrating a holy site.
But looking at this purely from a scientific perspective, I mean, even if she had been granted full access to that tomb, wouldn't any DNA test be fundamentally inconclusive? We don't have a reference genome for Jesus of Nazareth.
It is scientifically impossible to make that match. Even if an excavation yielded pristine first-century Middle Eastern DNA from that tomb, there is no baseline to compare it to.
Yeah.
You cannot prove a genetic match to a historical figure without a verified sample of that figure's DNA. Furthermore, a shrine visited by thousands of pilgrims for centuries presents the exact same contamination nightmare as the shroud.
Which begs a larger question. If we don't have a baseline for Jesus, what do we know about the genetics of the people living in first-century Judea?
Well, we know an incredible amount, thanks to a landmark 2020 study published in the journal Cell. Geneticists analyzed ancient DNA extracted from 73 skeletons unearthed across the southern Levant, dating back to that exact era. The data proved that the inhabitants of first century Judea possessed a very strict, distinct Levantine core ancestry.
Meaning they were biologically closest to modern-day Samaritans and Palestinians.
Yes. The historical Jesus, living as a Galilean Jew, would have undoubtedly possessed this Levantine genetic profile. He would not have had a South Asian genetic profile. Mainstream scholarship and hard archaeological genetics firmly place him as a biological and cultural product of the Levant. So, while the Rosa Bell theory is a compelling cultural narrative, the Indian DNA on the shroud does not scientifically validate it.
It is wild to observe the gap between what genomic science actually reports and what the public wants the data to mean. When the 2026 study dropped, societal reactions instantly polarized. You had regional news outlets in India celebrating the data as a massive point of national pride, running headlines like, "Even the shroud had to come from India."
Yeah. Which, is highly probable regarding the textile's origin, but the narrative rapidly warped into a claim about biological heritage. Conversely, skeptics and traditionalists wildly misinterpreted the 40% figure, thinking the scientists were making claims about the literal blood of Christ.
So what does this all mean? The massive disconnect here comes down to a fundamental public misunderstanding of what environmental DNA actually represents. In relic research, DNA is rarely the object itself. It is a recording of the object's journey.
The Padova researchers explicitly stated this in their conclusion to safeguard against these exact misinterpretations. They wrote that the shroud came into contact with multiple individuals, thereby challenging the possibility of identifying the original DNA.
But technology doesn't sit still. If next-generation sequencing gave us this massive, blended biological encyclopedia, what is the next frontier for relic science?
The frontier everyone is watching is single-cell metagenomics. Standard NGS is a bit like pouring 100 different fruits into a blender and tasting the smoothie. You know there is strawberry and banana in there, but you can't reconstruct what the original fruits look like.
Oh, that's a good way to put it.
Yeah, so single-cell metagenomics acts like a pair of microscopic tweezers. It isolates and sequences individual cells one by one. The hope is that one day technology will be precise enough to separate the manufacturer original DNA, like the skin cell of an ancient Indian weaver trapped deep inside the cellulose from the burial original DNA, the biological material the person actually wrapped inside the cloth.
That level of microscopic unweaving would be unbelievable.
If we connect this to the bigger picture, the shroud is undergoing a profound transition. It is expanding from being purely an object of religious veneration into a global biography. It is a single piece of woven flax where ancient Indian craftsmanship, deep Levantine history, and centuries of European preservation all physically converge.
It is a literal intersection of the ancient world. Let's recap the core takeaway for today. The 2026 DNA analysis does not rewrite the biological ancestry of Jesus of Nazareth, but it powerfully and undeniably rewrites the biography of his purported burial cloth. It strips away the simplicity of a medieval forgery and reveals the shroud as a deeply complex ancient Indian textile imported through vast, interconnected global trade networks.
It is a brilliant reminder that history is rarely as isolated as we assume. The ancient world was a highly mobilized, heavily connected place.
And I want to leave you with that concept of biological accretion of an object acting like a sponge. Think about the mechanics of this for a second. If a simple piece of linen can absorb and protect thousands of years of human trade routes, local diets, and shifting atmospheres, what invisible biological stories are the everyday objects sitting around you right now, quietly accumulating? What genetic barcode is your favorite jacket, or the coffee mug on your desk, currently saving for future archaeologists to find?
It absolutely makes you look at your physical surroundings with a lot more respect.
It really does. Thank you for exploring the interconnected ancient world with us on this deep dive. We'll catch you next time.